from the coffee, and we can have a few questions then afterwards. So the first speaker of today, for this morning's session, is Ana Diaz-Fernandez. He will talk about from SELEX to computational assisted optimization. So the stage is all yours.
Thank you. Good morning, everyone. I'm Ana Diaz-Fernandez from the University of Oviedo, and today I'm going to present you our work on targeting soluble BCMA with DNA aptamers from SELEX to computational assisted optimization. So I'm going to start with a bit introduction just for a little bit of context. Multiple myeloma is a blood cancer that starts in the plasmatic cells of the bones, and it consists of the uncontrolled proliferation of plasma cells in the bone marrow that generate abnormal production of immunoglobulins, giving an end organ failure. For this disease, B-cell maturation antigen, or BCMA protein, has emerged as a promising biomarker because this protein is overexpressed on the surface of plasma cells, and a soluble form identified in serum and blood open new opportunities for liquid biopsy. Nowadays, these sensors are based on antibody-based assays, and there is no DNA aptamer described for this protein. So our objective was the selection of the first DNA aptamer against this protein with high affinity and selectivity to be employed in the development of an electrochemical biosensor. To do so, we designed a SELEX targeting this protein. We characterized the selected aptamers, and we performed post-SELEX modification guided by computational modeling assays. So first of all, we did the SELEX. You all know this. So what we did was to perform a protein SELEX using magnetic beads. The commercial protein is a chimeric protein, so we employ magnetic beads modified with protein A. Then, after the interaction with the magnetic bead, just applying a magnetic field, we can separate the non-bound DNA, and then by applying hot water, we can easily separate the bound DNA to do the PCR and the separation to start a new round. Also, in all the rounds, we perform a negative selection against our proteins modified with protein A and IgG. But in this case, we took the not bound DNA to start the new round. So in this way, we perform 9 rounds of selection. Here you can see the enrichment that we have in each assay, in each round. We perform this with a binding assay with absorbance measurement. And as you can see, we start increasing the enrichment until the round 6. But then there is a decrease. So we decided to stop here and keep the round 6 to perform the cloning and sequencing. We selected 40 clones, and then using the online tool Cluster Omega, we classified them into families to identify the motifs for each family. And also based on the secondary structure, we selected 5 aptamers for further characterization. So to characterize the aptamers, first, we did empirical studies in order to know the affinity and the selectivity, and also then the computational studies to predict the 3D structures of these aptamers and to do some modifications to improve their characteristics. So I'm going to start with the empirical studies. So to do this, we perform electrochemical binding assays. In this assay, we have our target protein immobilized on the surface of gold electrode through a monolayer, and then our aptamer labeled with biotin. Then we have our conjugate, that is streptavidin HRP, and after the addition of TMB, it generates an electroactive product that we can measure. In this way, we constructed this affinity curve for the 5 aptamers, and as you can see, almost all of them bind to the protein. For example, this one doesn't bind, but we have very good aptamers, some of them with a nanomolar, low nanomolar affinity. Then we study the selectivity against human serum albumin because it's one of the most abundant proteins in serum, and IgG. And as you can see, some of them are very good, others not so good. So at this point, we decided, okay, let's see if with the computational approach, we can improve our aptamers. To do so, we employed a computational model, semi-automatic, that we call ActaMD, that we developed together with Professor Dima Suarez and Natalia Diaz, that is freely available, that all of you can use. Is here in this link, or if you scan this QR code. So what our model does is, starting from the sequence of our aptamer, just by having the secondary structure by MFOL, NewPack, or whatever other software, we transform it first into a 3D structure using, at that time we used RNA Composer because we couldn't find a tool to predict the 3D structure of DNA. So because we have RNA, first we have to transform it to DNA. So that's what this does. And then to relax the structure by applying dynamic simulation. So taking into account also the buffer and the ions. So at the end, we have a cluster or 3D model cluster, very relaxed and stable for our aptamers. So we employ this with 3 of the aptamers selected. The first one was BCMA23. This is the secondary structure. And as you can see, it has like a hairpin here, a small one, and a big one here. So we thought, okay, maybe we can improve the affinity by truncation, but based on this, it's almost impossible where to cut it. So we perform our protocol, and this is the structure. And as you can see, it only kept the secondary hairpin, and it's a very elongated structure. It's different from the one that we can think if we see only the secondary one. Also, if you look here at this part, this is these 5 kinins that normally we introduce them to separate our label from the aptamer in order that it doesn't affect the affinity. So here, when we add them, it doesn't affect the hairpins, but in the 3D model, we can see that they are affecting, they are stabilizing new contact. So we thought, okay, maybe this is the problem. And if we remove these 5 kinins, we can improve it. So we did so, and we tested them empirically. And as you can see here in the light blue, we have the aptamer with no kinins, and it seems to be better, to have better affinity. So maybe having here our biotin is affecting the binding. Then we move to our aptamer BCMA24, and at this point, this aptamer has multiple windings in its structure, so we thought maybe it has G quadruplex structure. So first, we perform circular decoherence analysis as a technique to confirm the secondary structure of this aptamer, because it has been described in the literature, the peak profiles for each type of structure. So we did so, employing our aptamer and also a control DNA that we knew it had hairpin DNA structure. And as you can see here, the profile of peaks corresponds with a G quadruplex structure, and moreover with a parallel orientation, that it may look like this. So here we have another problem that probably you all know, that drawing a G quadruplex structure or predicting the 2D is not that easy. So first what we did was using this software to try to know the one-ins of the structure that were involved in the different planes, then use the 3D NAS to build the first 3D model, and then our protocol to relax the structure. Using this, we came up with this model that, as you can see, we have here the 4 planes of the one-in quadruplex and 2 big loops. Also, based on this, we couldn't came up with a form of improving our affinity. So we moved to the third aptamer, that is the BCMA15. And as you can see here, this aptamer seems or is longer than the other ones, because we kept the primers of the PCR that are these parts in green. Normally, when we work with the aptamers, if the primer doesn't affect the secondary structure, we remove them because it's shorter, it's cheaper, and it's easier. But in this case, we didn't do it because they affect this secondary structure, so we kept them. But we thought, okay, let's see if we can cut them. So first, we try to remove the part of the primers that were not involved in the hairpins, having this secondary structure, and also in our model, we saw that both of the hairpins are kept. So this is a stable structure, and then we remove completely the primers, like here and here, keeping only the hairpin 2 that looks like this, and is a stable structure that we can, or it should bind to the target. So then we tested them empirically, and here is the result. So as you can see, we have a lower saturation signal, but if we look at the affinity constant, they seem to be better than the complete one. And the truncation this one seems to be a little bit better. So maybe meaning that hairpin 2 is involved in the recognition, but also we need this one to bind to the target. So just to conclude, we have developed a selection strategy for identifying aptamers directed against the soluble form of BCMA. And after 6 rounds of selection, we have obtained 5 aptamers that among them BCMA 20, 23, and 24 show the highest affinity and selectivity. Also, we have seen that the secondary structure of BCMA 24 reveal a parallel G quadruplex conformation, and also that traditional 2D models are not always reliable to predict post-SELEX modification, and that our protocol allows us to predict the influence of label and spacer in the aptamer structure. Also, before finishing, I would like to invite all of you to contribute to the special issue in aptamer-based biosensor that I'm co-guesting together with Professor Cecilia Cristea. So just to finish, I would like to thank my research group, the electroanalysis research group, the one that you can see here in the photo, and the financial support, and also all of you for your attention.
Yeah, thank you. Yeah, I mean, well, very good work. I just have a little bit of research visits with the G quadruplex. I work by fascia. We did be careful with interpreting the CDs particularly, and I mean, I saw that was a 4-layered G quadruplex parallel structures. It's nearly impossible to create that in reality, except with heavily modified loop. But that's for later.
Because it's not published yet.
Yes, but looking at the models, it seems close to impossible for you to have a 4-layered quadruplex.
This model also, instead of using this software, another one, and we lose one of the planes. But well,
but okay,
there are many G's in the structure.
So, but interesting about the MD itself. So I wonder, you start from M-fold, and apparently the structure goes very differently at the very end. So I wonder what happened with the input structures itself before doing the MD, and how long do you do the MDs to finally get that kind of structures? Thank you.
Yeah, this is in collaboration, as I said, with those professors. So exactly the time, I think it takes like 1 week or 15 days. I'm not really sure because they do that part. But yeah, that was surprising for us at first that it changes a lot. I think we do the molecular dynamics in the microsecond for 15 days, but yeah, I'm pretty sure it's that.
So my question follows on the CD. So I notice you have a sort of an extra hump on that CD spectrum that I wouldn't expect for a parallel G quadruplex, but it looks like it's coming where the anti-parallel G quadruplex should peak. I don't know if I'm right. I can't quite see the numbers, but so it looks to me like you've got 2 structures there.
Ah, okay. Well, we did it in another facility, so they analyzed the data for us, and they told us it's a G quadruplex with this orientation. So maybe we can.
You can get it a little bit more.
That
shoulder is something.
We know better how it's the structure. Yeah. Yeah.
Well, I mean, it's nice to see this because we do need structures in order to guide optimization. I think what you've shown here is that the tools that you use appear to be successful in improving things, but they do not necessarily validate the 3D structural proposal that you make. So I think some of these structures, I know you haven't developed the software, but I wanted to inquire whether the AptaMD tool, as it was published, whether they kind of also tried to determine the structures of aptamers for which the 3D NMR structure was already known, or whether some of these structures that you see here might actually be amenable to 3D structure determination, so that the value also in the structures that you get could be improved, or the validity.
But we didn't do it, but in the work that we published, we proposed different like 3D structures or one, and we said, okay, if we cut it here, it should work better or it shouldn't work. So we did some truncation and tested them empirically to see if the binding was better or worse to try to validate our structures. Yeah. So yeah, it would be something to do in the future. Thanks.
Right. So thank you again.
Put this here. I think that's it. Yeah, I'm from Sandtech, from Copenhagen. Yeah, so I'd like to thank all the organizers of this, and I'd like to thank actually a lot of you because you know, I've reached out to many of you when I first started this work, and you've been amazingly supportive. And it's really rare that you go into your scientific career and you're like, oh, these guys are amazing. So I'm really happy to be here and actually meet some of you in person. I've reached out over the years to get some advice. And so at first people were skeptical, but they're like, oh, maybe this might work. Okay, so you know, I'm here to talk about from computer to sensor and aptamer platform, combining in silico design, optical validation, and electrochemical detection of parvalbumin. So initially, I first started I've never actually done SELEX. I've never trained in aptamers. I did my PhD in biophysics, but in lipids. So I first started getting involved in aptamers because after COVID, I was like, why don't we have a detection device? We have the sequence of the structure of the protein. Why can't we use something? And I started looking into different ways to do it. Aptamers was one of these. So I started this, and these are some of the first papers I saw. And I was like, wow, these are really big numbers. You know, Andrew Ellington and Jack Choshak, Andrew Ellington at the University of Texas. And then, you know, Turk and Gold. I'm sure all of you have read these papers. And so you look at these really big numbers, and I like math. I'm not good at math, but I like math. And you look at the SELEX libraries, and the SELEX libraries are 10 to the 12 to 10 to the 14. But if you look at a random sequence of 4 to 60, it's actually one undecillion, which is a massive number. And you know, a number that's, you know, very difficult to comprehend. However, you know, sequence space does not equal structural space. So there's a lot of structures that are very similar. And like from your talk, you know, you might get, but you might also get some PCR bias and other types of bias in that. So like, okay, how can you actually sort of work on this? And you know, machine learning, we got a big computer to play video games. We're like, hey, we could use this for something else besides video games. And you know, Arthur C. Clarke said, any sufficiently advanced technology is indistinguishable from magic. And I'll tell you a secret, we actually don't know how these big transformer models work. We kind of think we do. You know, ChatGPT, you guys have all used ChatGPT, right? So yeah. So you know, we kind of know how they work, but we actually don't know how they work. And models, and then we're Oh, someone's recording it. Okay. Okay, cool. So yeah. So we yeah. So yeah, we developed these aptamer models, and we published a paper. I'll show you in a minute. Is it? I just yeah, it's still not going. We published a paper called Aptabirds, like Bert and Ernie from Sesame Street. We had this. Yeah. So, but we could use different models. And in this model, we'll use a variational autoencoder. So you look at this the probabilistic latent representation. So it's a very fuzzy way you try to sort of predict this, and we use a graph neural network to classify this. So you have the prediction, make a whole bunch of aptamers using the variational autoencoder, and we also use a mutational algorithm, and then we use a graph neural network to do this. And then in Scientech right now, we're using a lot of transformer models and diffusion, from inspired from the Baker Lab in Washington. So we have published Aptabird a couple years ago. We have somebody, Jesper Wengel from University of Copenhagen, and Danny Lang from Google, and then the rest of my team on there. And then we have this platform, which if any of you guys want to see during this meeting, or call me online, we have Aptimize, and I could show you how it works and how we classify this. And we're also making this better to see 2D, 3D structures. So the rest of my talk, this part we did in a couple of days, generating, classifying, figuring it out. And then we sent an Excel spreadsheet of a number of aptamers to our collaborators. However, so we're developing an aptasensor against beta parvalbumin, which is a fish protein which causes an allergic reaction, and fish allergies are really one of the top 3 allergies. They're present in bony fishes like carp and other bony fishes, but the thermostability of these proteins allow you to extract it quite easily. So this is a blueprint that we have in the paper. We took generated aptamers, classified them, then did the in vitro binding using fluorescence and a fluorescent quencher, and then we developed the aptamer sensor in vitro with a square wave voltammetry. So this is the part where we come in. We generated 6,213 aptamers. I don't know why we picked that number, but we started with this, and we just generated a number of aptamers, and then we filtered them down based on the dissociation and constant apparent, which is around 7.67 to 369 nanomoles. And then we have specific KDs we wanted to choose for, and there was a specific size that we wanted to choose for. The reason being is we looked at the papers that made the methylene blue, and these were sort of some of the parameters that, so the methylene blue aptasensors, these are some of the parameters that we found that were interesting. There's other ones, but so we have filtered those down to 100, and then down to 30, and the 30 we sent to our collaborators in Spain. And so we didn't. This is the last part of our work. We have a lab, but we didn't do any of the lab work on this. So we wanted to get experimental validation from our colleagues. Once we tested these, we actually did some sort of MD simulations and docking, and they seem to dock to a lot of positively charged residues on the surface. And 2 of these, I think aptamer 10 and aptamer 19 are very similar. They bind 2 similar sites on the, and then aptamer 29 is slightly different. So in order to, I think they tested all the 30 aptamers, and they used a 13 nucleobase capture probe, and with a black hole quencher, and then they had methylene blue on the other side. And then when the beta parvalbumin bound, it did the structure switching, and the capture probe left, and then you get an increase in fluorescence. So then we found that 3 of them worked really well. So aptamer 10, aptamer 19, and aptamer 29, at 7.4, you got this change in fluorescence. However, at the isoelectric points of beta parvalbumin at 5.2, aptamer 29 worked amazingly well. So they took that and they're like, okay, this is where we could go from here. And they compared it to a control and then beta lactoglobin. They found that beta parvalbumin it was much more specific than this. So, putting on a sensor, as Rob Batchelor and Nutromics likes to say, has to do a downward dog. I like to repeat this: so the methylene blue has to hit the gold in order to make that circuit. So we found that the aptar sensor works, and we found that the limit of detection is 3.3 micrograms per milliliter, and it has a pretty wide range. So they increased the concentration of the beta parvalbumin, and found that with the density that they used on the gold electrode, we found a very nice range. So, but making aptar sensor is one thing in the lab. Does it work in real life? So I actually don't eat any of these foods, but they look quite good. So they managed to purify these extracts and do like a Bradford assay, and they compared what they had from the Bradford assay to the EABC, and we got very good correlation between the different matrices. So the biosensor actually works, and so the collaborators, some of them work for these companies, or the part of the university that does testing on food. So they could actually use this in their tests. The paper would have been out a long time ago, actually, but we had some. We're trying to patent these sequences in this. So we could actually use this electrosensor for. So hopefully. So in general, we could generate sequences de novo. I could show you this with aptamers. And it could replace SELEX, not only for proteins, as we showed here, but also for small molecules. So we have both, and it could be for DNA and RNA. Aptamers can bind to distinct regions of beta parvalbumin. We're continually testing this. We have a platform where it goes from like 2D to 3D, and we use bolts 2, and then some nice docking platform using Brian Jimenez light dock. We also made Haddock, so we have a good docking platform. So we showed this using in vitro using fluorescence and square wave voltammetry, and then developed an app sensor that could work in real life samples. So yeah, that's basically it. I'd like to thank all of my collaborators. A lot of the in vitro work was done by Anna Abro from her PhD, and then her supervisors, Rui Campos and Monica, and then Marta. However, on my side, the Scientech team, George did all these simulations. Fleming did a lot of the work for the transform models, as well as Marco, and so we're happy to bring this to the world, make more sensors. Oh, thank you. Oh, a question?
Hi, thank you for the talk. So I was